This talk, so it's, the topic, phase transitions in biomolecular assembly. That's what I'm going to talk about, but it does relate to cellulose to a large extent. It's about how biological materials come together. And that is also, like Eero here started, we have a Center of Excellence, which is funded by the Academy of Finland, starting actually just beginning of this year, which is called the Center of Excellence in Life-Inspired Hybrid Materials. So previously we've been talking, for example, about bio-inspired materials, but the next step is then life-inspired, which means that we are trying to get more lifelike properties into materials. Lifelike properties are things like adaptability, self-repair, ability to change in conditions, and so forth. And that really builds then on understanding a lot of biological processes beneath and how materials function in a deep way. Okay, so. And one of these functions is, and that's very much then in the topic here of this seminar on interactions and water interactions, is about adhesive interactions. Let's see how this works here. Adhesive interactions between materials. So here what you see is like a part of an old dock. It's probably been in the water for around 50 years. And what you can see is this barnacle sticking to this cellulose, to this wood. And it's quite interesting. So that has been formed in water. And for those of you who are boat owners, you know that these are extremely hard to get off the hull of a boat. So the interaction is very strong. This happens in water. It's very stable, and it forms an interesting then inspiration for looking at that. What is going on here, and for what kind of applications? How can we use this in kind of a materials for materials applications? Because really, what this is about is then maybe is finding ways of extending the use of natural materials like cellulose, so that we use, water-based chemistry is clear. Our materials are in water. Water is an idea. It's a very, very good solvent. It's definitely ecological compatible. But the thing is that how do we then work with interactions in water and still forming in water, but being, for example, then stable also in water. And overall, how do we build materials in the future that are completely ecologically compatible? That means that during the formation, the use, the raw materials we have, if they're accidentally come out in nature, in the ecological systems, so how can the ecological system still take care of them? Those are like driving ideas behind this research. And so there's been a lot of studies on marine organism adhesives, blue mussels, how they stick to surfaces. That's also a case of interactions forming in water and being stable in water. So these are blue mussels here, but another interesting part is breaking these mussel shells apart. And this is a picture taken by my former PhD student, Peshman Mohammadi, who was doing electron microscopy on different structures. And what is then a reoccurring theme, both when it comes to the adhesives that mussels form, but also then within their shells, is what you can see a trace of here, that using fresh shells, the adhesive material there is not a dry, brittle one, But you can actually see, it's maybe not completely clear, but you can see these kind of threads, and then this kind of liquid forming here. It actually seems that it's in a liquid form, this material, this adhesive between what is then the calcium carbonate crystals there. And this theme about how adhesives form is then already becoming quite widespread. And it's also understood that the barnacles that I just showed in the previous slide, their adhesive mechanism is also based on this the form of this adhesive, which are proteins. They're liquid forms. And here in the next slide, I'm showing another type of biomolecule, which is a recombinant silk protein. But these adhesives that are found in barnacles, within the shells of blue mussels, and also the blue mussels used to adhere to stones and other structures, they are of the same general type of proteins as silks, and we call them they're generally a silk type of protein. What we're doing here is we're working with silks that come from spiders. And in a certain, so what you can see here is it's a beautiful video about on a system that has phase separated. Now I'll be talking a bit more about that. And it looks quite special when this system is flowing there, where you can see the reason why these structures are so elongated when they're flowing is that they have an extremely low surface energy. So there is no energetic cost for extending these structures. These are formed spontaneously in aqueous solutions. And typically what you can do when you study these proteins, and these are then the silk proteins that we're working with, so you can have just increasing an amount of protein, and then you do something. And in this case, what we did was adding dextran. And dextran doesn't interact with these proteins. It just kind of crowds the system. So it kind of competes with water, and it kind of pushes these molecules towards each other. And then that will cause these proteins to form these liquid droplets. So they will phase separate spontaneously. And then the region there, which is shaded in gray, will look something like this, that you'll see these liquid droplets forming. And then you'll see here in concentration, so going then above this about 100 grams per liter, it will form just spontaneously without any crowding system. The other things that you can do instead of crowding with the dextran, that could be maybe a change of pH, add of salt, or something like that, that would drive these proteins together. So, so here is a video that now is a bit tilted, so you can't really see the full video as it was intended. So it's supposed to show a droplet and this microsuction needle. You can see here, it's actually in a funny way distorted, this figure. Where you can see there, it's not supposed to be distorted. It's supposed to be completely symmetrical. And you can just take a needle and then pull on these droplets. Will, for example, allow you to characterize physical properties like surface tension. In this case, it's actually below 30 micronewtons per meter, which is a very low value indeed. Even below 1 is a really low surface energy. And what does that mean? Well, actually, I come to this slide first to describe a bit more what our proteins look like. What type are these silk proteins that we use, and like in general. So in general, also the proteins that functions as adhesives, they are non-structured proteins, intrinsically disordered proteins, which means that they don't take a three-dimensional fold. We'll see here on the side here, you'll see actually parts that do take three-dimensional folds that are here on the yellow structures. Those are terminal domains that we have added, and they're actually added there to bind to cellulose. But the middle part doesn't fold into a three-dimensional structure in normal conditions. And here is the sequence then below. It actually is a repetitive structure consisting of 12 of these types of units. What I've showed there in blue is a stretch of alanines. And alanine is the amino acid just has a single methyl group. So it's a hydrophobic amino acid. And this contains this kind of repetitive structure, and that's typical also for intrinsically disordered proteins to have this. And you can see here a computer simulation of these proteins, and you see that the stretch here, which is blue there, the alanines, they actually take the shape of alpha helices in this protein. So these simulations were done just to see how these proteins behave in an aqueous solution. Then putting a lot of these proteins together in a simulation actually resulted in them forming a network structure, which are connected both by a weak adhesiveness, a dimerization between these terminal domains, but then something that is called, actually in the polymer field, is called stickers, because these hydrophobic units that I was talking about, these alanine stretches, they have a weak interaction with each other, and they will stick to each other. So it forms a network which you actually then can see also by cryo-SEM. So that picture, this is here, this is an actual cryo-SEM image of a fractured droplet, and then the computer simulation actually suggests something similar. And this then also allows us to understand that this thing about surface energy, that what keeps these structures together are weak interaction between these protein polymers. They're not stabilized by interfaces. They're not stabilized that you would stabilize lipids by surfactants. There's no surfactant there. The interfaces don't need to be stabilized. It's just another state of the protein, which is condensed. Okay, and then that leads to some interesting consequences. And this image here, which is then scanning electron micrograph, is what happens when these proteins are just take a solution and dry that down on a film and slightly stretch that film and then image. So what you see here are these traces of these protein droplets on the film. There's something funny with this laser. It kind of keeps on turning itself off. So you see those droplets, you see the traces of them. And when a crack where that was extended, going through here, and there's a zoom up of that, this really requires some skill, this thing. What you can see is that these threads bridging this crack, and that's called, in polymer field, is typically called crazing. And that comes in polymers typically from I'm just reading here, the note says, please stay close to the microphones. I hope I'm okay. Not too close, because I was bumping into it. Okay, so yeah, so in the polymer field, usually this means that the polymer chains are entangled. So there's just a figure for you to kind of imagine how these would be sticking together. Possibly other interactions. There are interactions between the polymers that might be doing that. Yeah, I'll come to that in the next slide. But really, what this what this shows us very intuitively is that the state of the protein in this condensate is they're in very close contact. And they're in a way, not close contact in a brittle way, they will, like a crystal, but they're in close contact in a way which they still can slide in relation to each other. And that is what we think is a key thing for how to use them. or for their properties. So another thing, just briefly, is that I was saying that the alpha helices, so there's actually a lot of detail to this, and I won't go through all the detail. I didn't even check the time now, how much detail I can do. So the alpha helices actually tend then go to beta sheets, which are lower energy levels, and when we can do that trick kind of to solidify these proteins also still further. By then a lot of different tricks like just over time, specific ions, solvents, and so forth. So there's kind of more dimensions to this actually that I'm going to talk about more. But now we'll come then to cellulose and why I told you this story about how proteins behave, typically adhesive proteins behave. And we heard that from Orlando already with his paradoxes this morning. And one of the paradoxes, I think, was like, why take things apart? No, it wasn't really, it didn't go that way, and he's not here now. Well, anyhow, I'll just try to improvise something about taking things apart and putting them together, right? Okay? And here, this is a picture from the lab of Ingo Burgert in ETH, where they are just saying, if you know, why take it apart and you know, put together when you don't have to take it apart in the first hand? So what they do, and a lot of other people also, of course, do, is delignify cellulose. You'll keep the structure, but then you're able to work with that structure in different ways. So what they did was they compressed this structure, and that gives very strong materials, high stiffness and strength with compacting, so you squeeze them. And really, one of the reasons why to work with this also actually was also said already by Orlando is that we need to figure out ways to work with cellulose over the lifespan. So how to recycle cellulose when it's degraded into smaller and smaller and smaller chunks. How can we kind of upgrade it after that? And this is one way. So if you're going to delignify it to make it a strong material from chunks, you have to be able to glue it back together. And this is then how this silk comes in. Okay, this is just quickly then a slide in between. How you do that, you heat it in acetic acid and hydrogen peroxide, and then you'll get rid of the lignin. And what we did then, and I'll show here first, so we actually then used these proteins that I just told you about earlier to glue these pieces together. And this is what the delignified cellulose looks like. And just for a technical reason, so there's like these plastic sheets you see like there's a gap in between, and there's one sheet beneath this, and then there's this gap, and then in that gap, when they're on top of each other, you can't see the sheet beneath the plastic ones. So there, our adhesive is put, and then it's compressed with a vacuum suction, and then it's all experiments then are of course done at a controlled humidity, which is 65% at this time, because that's what we have facilities for. Okay, and this is the result from just lab tests, then shear strength tests. And then illustrating here, you got the two sheets of cellulose, the adhesive in between, and this is the result. So for silk, the protein that I was showing, the computer simulation and so forth, had actually quite a large spread. So we could come even up to strengths of 10 megapascal, that average around 7 megapascals, but some samples being actually surprisingly and disappointingly weak. So as a reference then, we used one reference is starch, and that's also what they've been doing then in Ingo Burgert's lab, using starch as an adhesive for this system, which is of course a polymer that's a large polymer and a quite kind of stiff polymer. And then another type that we used as a reference is bovine serum albumin. So BSA is kind of the general protein. If you want a protein, like as a reference, you take BSA. And BSA is in most cases a kind of a bad reference because it's an extremely sticky protein. But as an adhesive, then that's why it's kind of interesting to look at. And in many cases, yeah, it actually functions quite well as an adhesive. So a lot of these type of traditional glues even contain albumin as a component, like animal protein-based glues, so bovine serum albumin. And that was a decent adhesive. And then water. Water means that we just don't have any adhesive at all. And as we all know, cellulose will stick to cellulose quite well. And this is actually not so bad, the result, with just actually having nothing there. So, but then we try to figure out what is the reason why we would have such varying results with the silk samples. And then, which is not, it actually cuts off my slides a bit here. So then Jenny and Laura, who were doing these experiments, they observed actually quite an interesting thing, is that for all the samples with the silk that were weak, we found a lot of tear out when in the adhesive, in the lab share test. So they would kind of rip off the surfaces. And all the ones that were strong, they were clean. So they're very clean surfaces on that. It's kind of initially counterintuitive because you think like if it's a lot of energy to rip off structures, that would be the strong ones. But they were always weak, the ones with a large tear out. So then looking at those references that we had, like just water, we saw like intermediate tear outs, a bit of cellulose coming off. BSA was always very clean how it fractured. There's no rip-off. And starch always kind of a lot of rip-off, like a tear out in that rip out in that system. So this is actually the only, or one of the main then kind of, we then wanted to, we kind of didn't really understand this, the logic here, that on the one hand, silk samples were sometimes showing low tear out for strong ones, high tear out for weak ones, but that's not corresponding to the other ones overall. So that was, again, BSA, no tear out, and then with more or less similar strength, starch had a high tear out. And so we analyzed samples to still try to understand them further. One thing that comes to one's mind quite naturally is that with adhesives that you should put more glue, the better. Like if you want to glue something, you should put a proper amount of glue. According to literature, that's not the case. More glue is not better. And that actually is something that we could verify here. So this is then by amino acid analysis done from these samples, exactly only the glue area. Whoa. This is not my intention there to change slides. Okay, so for the square ones, there are BSA. So you can see it sticks not as much as the round ones, which are silk. BSA sticks a bit worse than the silk. I'm trying to figure out how this works, the pointer. So, but on the other hand, for the silk also, you don't really see a correlation between strength and amount. And in this system that we have, they just stick as much as they stick because when we do the vacuum suction, it's in a wet system. It will actually take off kind of the surplus one. And then the other one here is strength and stiffness. So there's like an overall, you can see the silk ones, which are the red ones there, they're always a bit stronger and stiffer, and there's some kind of weak overall correlation in how they work. So, but the one kind of general misconception about these lab tests is that it's a general test that it will tell you it's a comparable system or comparable results between systems. But that turns out that that's not really true. And one of the reasons is that, as you can see here, I think this is not a video, but this is the way they actually were tested in the tensile tester, the cellulose samples. And here, a bit exaggerated, is what you see is that there's a bending, because when we put them in the system like this and it pulls, then of course they, you know, the this part tries to align with that part, but it can't do it because they're kind of overlapping, so it has to bend. And we can see that quite clearly, that these samples bend. And this is one thing also that leads to variation a lot in this type of tests. Because one thing that happens here is that because there's still some extension of the cellulose, and that will extend a bit more when you here where the force is higher, and then like the corresponding way on the other side of the adherent. So there's going to be an unequal distribution of stress. And also because they bend, you'll have more kind of normal forces. So it's actually tearing apart more. So this will create what is known then as a deformation zone in the sample. And generally also it's known that if you're doing lab tests, so if you have a very strong adherent that won't bend at all, then you'll get almost anything to stick to that system. So, but in a system with a lot of deformation, there are more things happening, and this is the way we then can try to explain what's going on. So, the entanglements that I was talking about will create a strong interface. If they go over the interface, that will be strong. Also, if there is a good infiltration in the system, that will give strength. So if we take the example then of the starch, that doesn't infiltrate into the cellulose. So even though it binds well to the surface, it doesn't infiltrate to the cellulose, leading to a large tear out when it breaks. The BSA goes well into the system, but it doesn't create entanglements over the surface. So it will give us quite good strength to the sample, a clean surface, because there's no because it just goes in, it doesn't cross the interface. And then the silks, they were much stronger than the BSA. They infiltrate, but they can entangle over the surface. That were the strong ones. And then, presumably, the silk ones that were weak and a lot of tear out, they, for some reason, were not able to go into the cellulose structure and create like that type of strengthening of the cellulose structure itself. So, yeah, and this is, if you're interested in this story of adhesion, this discussion is largely based, actually, and the insights about tear out and using that on that book by Stephen Abbott. So, conclusions really about like molecular architecture and how to design proteins for getting the correct adhesive function. We find the inspiration then from silk proteins to understand this and actually build quite good systems that we don't still know quite well enough so we could control it actually. So we have still problems with the large variability between samples. Okay, and then I'll come into the last part, which is then collaborators and funding. And here you see a group of people in my lab who mostly worked on this, and then our various collaborators that help with the other results. And then this was actually part of, most of this work was part of the forest value project, funding project, was supported by the Academy of Finland in Finland. So thank you.